Okay, thanks, Erica, for the introduction. So I'm Jonathan, I'm a graduate student in Adam Martin's lab over at Building 68, and I'm going to talk to you today about eggs, basically. So egg cells are enormous, at least as cells go. So I'm going to show you a few cell types here, and these are all to scale, starting from, come on, a red blood cell and a skin cell, which decided to show up at once, and then a human egg cell. So this is already much, much larger than a skin cell. And then because our lab works on fruit flies primarily, I'm going to show you fruit fly egg cell next. So these are even huger. And one of the obvious questions that comes up here is how these cells actually get this large and why they don't stay at the size of skin cell, for example. And they need to be this big because egg cells are going to go on to support early embryonic development. So they need all kinds of the goodies that cells need to survive, or that organisms need to survive. So we set out to look at basically the mechanisms for fluid transport that helps these eggs grow. And fruit flies are really cool about this because for fruit flies, it takes a village to raise the egg. So typically, or what always happens is you have one egg cell shown here in magenta, connected to 15 support cells in this tree-like structure. And there's a whole lot of different choreographed steps that go on here, but towards the end of egg development in a process that's called nurse cell dumping, hence the title, very rapidly over the span of about 90 minutes, these 15 support cells transfer almost all of their contents into the egg cell, basically shriveling up and dying so the egg cell can survive. And what this looks like live using a membrane marker here, I'll show you in a second. And I want to point out two things as this is going. So first, these cells begin shrinking and they do so without really changing their shape. And then about now, you can see the cells start to wiggle. So these are pretty heavy cell contractions mediated by proteins exerting force locally. So we wanted to determine kind of the dynamics of this dumping process. So in what order and what direction and at what speed the fluid moves. And to do so, we collaborated with Jorn Dinkel's group in MIT Mathematics to try to model this as well. And kind of as a side bit of this, we noticed there's this protein called PCNA that normally exists in the nucleus. You can see it here as these cyan balls. And it actually gives us a hint to when the cells transition from the shrinking to this wiggling type of fluid transport. So this is another movie, and I want to point out, you can already see it in this cell, that the PCNA signal has gone from the nucleus to the cytoplasm. And in the same order that it does that, with roughly a 20 to 30 minute delay, the waves begin in the cells and they start deforming. So this protein's kind of neat because whether or not it's directly causative or anything here, we're still not sure. It does give us a sort of timer to let us know which waves are going, or which cells are going to begin waving and roughly when. So while imaging this one day, I had 4 of these that were relatively near together, and I just kind of pushed them around in the dish to get. Huh? Nope. There we go. Push them around on the dish to get them arranged like this. And something I think is really fun about this image is knowing what I told you about PCNA, you can tell the age of these egg chambers and how far each one of them is into the dumping process based on whether the PCNA is entirely in the nucleus, like the youngest one at the top, or mostly out of the nucleus, like the oldest one at the bottom. Something I think is cool about this process, besides just this being an interesting way that biology gets things done, is that when we were collaborating with Jorn Dinkel's group, it turns out the fluid flow, at least in part of this process, is very nicely described by a fairly simple physical law. And since physical laws are universal, it suggests that this type of fluid transport could likely be seen in other organisms. And in fact, mouse oocyte development seems to do something at least similar involving fluid transport between cells. Once PowerPoint decides to cooperate, I'd like to thank all of my collaborators here. There we go. Okay, so first of all, Yasmin, she took most of the video, well, both of the videos that I showed you and a lot of the images for this project, and she also trained me to work on the system. And then Adam, my PI, and Jorn and Nico, who did the theory and simulation for this project. And then everybody else in both groups for their wonderful feedback and also our funding sources. So, thank you.